All right, I would like to welcome everybody in today's Evolution at Vanderbilt seminar series. It's my great pleasure today to be the host because for the first time in our seminar series, we're hosting two trainees, and you'll hear about all their wonderful work. But I do want to encourage all trainees as well as their advisors who have interesting stories in their works. Please contact us if you're interested in presenting your evolutionary studies related work to our seminar series. We have some slots available in the spring. And so if you're interested, by all means, please contact us and we'll figure something out. So today is my great pleasure to introduce to you two graduate students in the program of biological sciences at Vanderbilt University. Both of them have done really cool work that has ended up on the covers of different magazines, different scientific journals, and so that's really exciting. First will be Stephen Ferguson. By means of background, Stephen got his undergraduate degree at the University of Colorado, Denver, and then he was recruited here at Vanderbilt, and he works in the Zweibel laboratory. And he'll tell you all about chemosensory regulation of collective behavior in eusocial ants. So, Stephen, thank you for agreeing to give this talk, and we look forward to it.
All right, am I on? Let's go. Good afternoon, everyone. I'm Stephen Ferguson, and today I will be talking about ants, which I think are just these endlessly fascinating little creatures. So ants are one of the most evolutionarily successful insects on the planet, and I base this argument on three observations. Ants are globally pervasive. They're found on virtually every habitable continent on the planet. They comprise up to 25% of all terrestrial biomass, and there are over 10,000 trillion ants on the planet, which amounts to about 1.5 million ants per person. So to put this enormous number into perspective, I want to provide a quick visualization. So here, each box represents 100 million individuals. So we see the total number of humans on the planet in blue compared to cattle and chickens, and then a small fraction of one box for elephants. What about ants? Well, first we have to scale this image down. So each box in the middle is now going to represent 10 billion individuals. And we'll do that one more time so that each small box now represents 4 trillion individuals. And you can see our original graphic with humans and chicken and cattle has been reduced to about the size of 1 pixel. So here's our pixel with all the people in the corner here. But what about ants? Here are all the ants on the planet. This is what 10,000 trillion ants looks like, and they clearly dominate relative to humans. Ants are going to take over the world, or they already have. So what makes ants so successful? I believe that their intricate social behaviors may be a key to understanding the success of these very wonderful insects. So through the coordinated behavior of many individuals in a colony, ants are able to achieve impressive feats, such as the construction of nests made out of leaves using the silk that they weave from their larvae. But they also engage in cooperative nest defense as well as food search and retrieval. And what makes these feats even more impressive is that they are able to accomplish these tasks without any form of centralized control. So neither the queen nor any specific individual within a colony calls the shots or coordinates the behavior of subordinates within a colony. Therefore, we can think about ants as complex adaptive systems. So here, complex behavioral patterns emerge from the interactions of many relatively simple agents interacting with one another. And these patterns are difficult, if not impossible, to predict when you examine the behavior of the agents alone. And these emergent behaviors then, in turn, influence or feedback onto the behavior of the individual agents. So ants, for example, may share local information about the location of a food source using chemical cues. And then through these interactions, complex patterns like foraging and the creation of these fungal gardens emerge. And this might seem like a very foreign concept to us right now, but humans also interact with one another, and similar complex patterns emerge from these interactions, things like cultural landscapes. Unfortunately, these patterns are not always beneficial. So for example, misinformation about vaccines or the effectiveness of wearing a mask may spread among these agents and produce aberrant outcomes. So by studying ants, we may learn something more fundamental principles that underlie a variety of different complex systems. So things from biology to mathematics to the social sciences and beyond. And so today I want to pose the question, what mechanisms are involved in generating the complex social behaviors that we see in ants? And the chemical cues that pervade ant colonies provide a clue to the answer. Chemical messages are extremely important in ant colonies. They provide a mechanism to both store and share information. And perhaps the most important chemical cue are these cuticular hydrocarbons, which provide information about colony membership, as well as the age and task of an individual. So how are these chemicals detected? In insects, there are three primary classes of chemoreceptors: odorant, gustatory, and ionotropic receptors. And these receptors are expressed on the dendrites of neurons, which can be found inside these sensory hairs, which we call sensilla along the antenna, as well as other chemosensory appendages. Odorants then enter into the sensillar lymph through pores along the cuticle, which are indicated by these black arrows in panel A. And in some way, these odorants are thought to be chaperoned down to the neurons. In dipteran species, such as the fruit fly or mosquitoes, each sensillum may contain maybe 2 to 4 neurons. However, in the ant sensillum, there may be over 130 neurons. So there's this tremendous complexity that we see in social insects that we don't really see in solitary species, even at this very basic level of the sensilla. So today, we're going to be focusing down on the odorant receptor family, which detects pheromones and other general odorants. And the reason we're interested in this family in particular is that odorant receptors in the genomes of ants and other social insects have undergone this massive expansion, characterized by rapid gene gain and loss. And this is something that we don't see in dipteran, for example. And in fact, ants and other social insects like honeybees have among the largest number of odorant receptors in any insect that we have described to date. And when we took a closer look at the OR family, one of the clades that immediately stood out to us was this Hymenopteran-specific 9-exon subfamily, which I have highlighted here in that red circle. And on the right, I've listed the total number of OR genes annotated across these different species. So we have the jewel wasp, we have honeybee, and then we have 4 different ant species. So it was tempting for us to speculate that the evolution of these odorant receptors may have facilitated the evolution of more complex social behaviors by enabling these ants or providing them with the tools that they need to communicate a broader range of information. So we've seen a massive expansion of odorant receptors in ants, and the 9-exon subfamily was of particular interest, but we had not yet identified the ligands for these receptors. So here we use the UAS GAL4 system to drive expression of an ant OR into Drosophila. And we were able to use electrophysiological techniques to determine which odorants each receptor detects. So we have heat maps with different odorant receptors along the X, different hydrocarbons along the Y. And we were able to demonstrate that the detection of hydrocarbons occurs broadly across these various OR subclades, including, but not limited to, the 9-exon subfamily. So we've seen a massive expansion of odorant receptors in ants. We've now demonstrated that they are responsible for detecting hydrocarbons. And the next step was to link the molecular physiology with social behavior. So we decided to look at nestmate recognition, arguably one of the most important and well-studied social behaviors. And in this system, ants from different colonies possess subtle differences in their cuticular hydrocarbon profile. And the detection of these differences leads to aggression between non-nestmates. And we have a very nice image that was taken by one of the Vanderbilt photographers of a couple of our ants fighting. So to begin, I wanted to establish an aggression bioassay. So here I would take two ants, either nestmates from the same home colony or non-nestmates from different colonies, place them into a petri dish with a divider separating the two in the middle. And we would allow the ants to acclimate to our handling before removing the divider and then recording their interactions for 3 minutes. These videos were scored by 4 separate blinded observers, and then we calculated an aggression index. And you could see one of our videos here. So we began by taking a relatively simple approach at first and began with ablating the antenna. So as expected, we found that non-nestmates in our sham control with both antennae intact were significantly more aggressive than non-nestmates. And this was also true in our unilaterally ablated ants, where we removed either the left or the right antenna. However, bilaterally ablated ants had a significantly reduced level of aggression between non-nestmates. And as a control for these studies, we also recorded some basic parameters of mobility and found that antennal ablations do not significantly incapacitate these individuals, and they don't, for example, outright kill the ant, which would not be very interesting for these results. So this was all well and good, but we really wanted to examine odorant receptors using a more targeted approach. Now, if you're at all familiar with the research done in our lab, you know that we were involved in generating some of the first genetic knockout ants using CRISPR. So we successfully knocked out the odorant receptor co-receptor in Harpignathos saltator. However, we ran into a problem, and that is that orco mutants experience these very profound neuroanatomical defects in the antennal lobe during development. Namely, there is a significant decrease in the volume and the number of glomeruli. So we can't determine whether the changes in behavior that we see are due to the loss of OR signaling or any number of changes that they may have encountered during development. However, we are very fortunate to have a powerful and, in my opinion, superior alternative to genetic editing. So using these VU class actives, we are able to manipulate OR signaling in wild type adults. So typically, odors will interface with the odorant receptor to allow the influx of ions into the cell. However, our VU class actives interface at an allosteric site on the odorant receptor co-receptor to either activate in the case of VUA4 or inhibit in the case of VUANT1 OR signaling. And while it's not pictured here, it's important to note we also have a chemically inert analog, similar in structure to VUA4, but not physiologically active, which we use as a control. So after some trial and error, we found volatilization of these compounds to be an effective means of delivery. So here we were heating these compounds on a heat block, and then we used compressed air to deliver the compounds either to our electrophys prep or to our behavioral bioassay arena. And we were able to show robust dose response curves to decane and 4-methyl-3-heptanol in both our blank control as well as our inert chemical analog control. And as expected, VUANT1 significantly reduced responses even at our highest concentration. However, VUA4 gave somewhat unpredictable responses that were nevertheless different from our control. So we then integrated this, I'm really tripping up on that word. We then integrated this volatilization paradigm with our aggression bioassay and found that these VU class actives did not interfere with the parameters of mobility that we had measured before. And we examined this both before, during, and after exposure to these compounds. However, both our orthoagonist and our orthoantagonist significantly reduced aggression between non-Nestmates. And so this was a very exciting result. However, Patrick Abbott very poignantly suggested or proposed a very important question for us, and that is that, are we interfering with aggression-mediated Nestmate recognition, or are we simply disrupting all forms of aggression by pacifying the ants, if you will? So we ran one more control and examined aggression towards a chemically neutral stimulus. And we found that disrupting OR signaling does not interfere with all forms of aggression, suggesting that ORs are necessary specifically for Nestmate recognition. So these results ultimately led us to propose this lock and key model for Nestmate recognition, or perhaps more accurately, non-Nestmate recognition. In this system, our orthoantagonist is analogous to a broken key, which does not activate any of our odorant receptor tumblers and therefore does not unlock aggression. Similarly, our orthoantagonist, our orthoagonist is similar to a rake, which would bump all the tumblers at once. But this is also not sufficient to unlock aggression. Rather, we need a precise chemical signature key, such as the very subtle ratios of hydrocarbons along the cuticle, to unlock this aggressive behavior. In other words, ants are asking questions first and then shooting later. And this restraint may be a very important component to their success. In a colony of thousands of individuals, you have far more interactions with your friends than you do your foes. And if uncertainty led to aggression, then the colony may ultimately crumble into disorder. So with that, I'd like to thank everybody involved in these research projects, and thank you all for your time and attention. I'd be happy to take any questions, which I believe Antonis will field, and then I'm also happy to talk more about why I think ants are so great in the reception later on.
Great, thank you, Stephen, for a wonderful talk. Everybody's welcome to submit questions through the Q&A function of our Zoom call. I'll start with a question. Maybe you said it and I missed it, but can you tell us a little bit about the origin of hydrocarbons, right? What is the source, right? So are they bacterial in origin? Do the ants produce them? How, what do we know about the cocktails of hydrocarbons they produce?
Yeah, so it is a combination of different things, beginning first with the queen. Will in some way sort of set in motion what hydrocarbons end up on these ants, and then through interactions among the ants within the colony, such as trophallaxis, where they're feeding each other, they help to share these hydrocarbons. So there is sort of at the front of their head, something called the postpharyngeal gland. It contains a similar mixture of hydrocarbons that you would find on the cuticle, which is thought to sort of get integrated maybe with some of the food that might be in the crop that's shared among ants. And then their diet could also influence these hydrocarbons as well. And environmental conditions, for example. So humidity levels as foragers go out and search for food, those types of things also slowly change the hydrocarbons. And in addition, and similar to humidity, for example, changes in weather and time and these types of things will ultimately determine what that hydrocarbon profile looks like. So it's a very complex landscape, and it's impressive that these ants are able to make those distinctions.
So if I was to follow up, so you showed the videos, which are really beautiful and very convincing about, what is aggressive behavior and what is non-aggressive behavior. Can you give us a sense of how quickly, right? So they recognize the odorant receptors recognize the hydrocarbons, and then there is sort of a response. How quickly does it play out? And do you see, as you compare, different strains or different species of ants, do you see sort of different speeds of reactions?
So towards the first question, aggression seems to happen almost instantly. If the ants are going to fight, that first contact that they make with each other will likely lead to aggression. And if they're not going to fight, then probably they're not going to fight in a minute from now or 2 minutes from now. With the exception of if they were recently disturbed, and that's why we give them some acclimation time, and that can influence aggression in that way. The second part of the question with different species. So I'm not sure about reaction time. It would be interesting to see if certain species or even certain castes within a colony have different reaction speeds, for example. So in Camponotus, the ants we're studying, we have smaller minor workers and larger major workers. And some people suggest that maybe those are a soldier caste. And certainly in army ants, there are very large morphological differences, and we may see differences in reaction time that way. There are interesting differences between species in terms of whether they actually engage in this aggressive behavior. So I talked about Harpygnathos saltator and the problems in the brain sort of limiting our ability to use those ants for nestmate recognition. But what I didn't say is that those ants also don't really engage in nestmate recognition. So ants of the same species, they will sort of welcome into their nest. And that presents some other interesting things to consider when looking at aggression.
Great. And then my final question was, you mentioned that you use the VU class actives instead of the CRISPR-Cas genetic modification, and that this mechanism or this way of assessing behavior was arguably better suited for the study. So can you expand on this? Why do you think agonists, because it's a classical technique, right, compared to sort of the new fancy CRISPR assay. And so I was wondering if you could elaborate on that topic.
Yeah, so there's a few different reasons why we went that direction. The first is that the genetic knockouts introduce all kinds of different issues that might come up during development that would change behavior that aren't really related to OR signaling directly, but rather some event during development. The other issue is that getting CRISPR to work in ant species is very challenging. The reason that CRISPR worked in Harpygnathos is because we could take advantage of their very unique reproductive system, where when the queen of those colonies dies, the workers will engage in this dueling behavior and establish a new reproductive. And we can take advantage of that by isolating workers and creating as many reproductives as we need. However, in Camponotus, the ant species that I was studying, once the queen is dead, the colony will in time die as well. And so you have all of these eggs you could inject, but they're all going to develop into sterile female workers that don't propagate any further. And so because of that, you would be able to maybe be successful with CRISPR, but you would never establish a line. And so there would be this tremendous amount of work. There's interesting ideas to address that with things like remote control and maybe some other paradigms that are going to come through. But for now, it's very difficult to get those up and running. So for the pharmacological compounds we used, we could actually have wild type adults that had, you know, sort of a normal developmental trajectory, even though they're in a lab. And then we could manipulate OR signaling at a very discrete point in time and look at behavior then.
All right, great. I don't see any additional questions, so thank you so much, Stephen, and we'll move to our next talk.
Great.
Thank you again. So it's also my great pleasure to introduce to you Jacob Steenwyk. Jacob is a graduate student also in biological sciences like Stephen. He's working in the Rokas lab, in my lab. Prior to joining Vanderbilt, he was a undergraduate and then a master's student at Clark University. And he's currently an HHMI Gilliam Fellow. So Jacob, take it away.
My apologies, I forgot to unmute myself. Okay, sounds good. Thanks, Antonis, for the introduction. Very happy to be a part of the Evolution Seminar series. I realized since this is going on YouTube that I should probably take a moment to introduce myself and say a little bit about me. So there's a couple ways to link up with me and see what I've been up to. I have a personal website and a Twitter. These are probably the best ways. And on my website, you'll get broad information about the research that I do, links to certain software that I've developed that could potentially help your research. And also on Twitter, you can find very random facts, seemingly random facts about diverse fungi, as well as some science-themed art. And the theme that ties all of this together is really this love and this passion for biology. And that's why I choose to study biology. It's fueled by this love for biodiversity. And I express this through art, science, and communication and outreach. And to just say a quick vignette and highlight the arts and sciences, I want to acknowledge a new shop that I've made called the SciArt Shop, where different organisms are featured in their portrait form on vinyl stickers, poster prints, camper mugs, and most importantly, 100% of profits go towards global conservation efforts. So it's really purchasing with a purpose. So just to show you some of the portraits that I've featured in this shop, a lot of them are endangered. So the African wild dog is an endangered species, as is the Galapagos penguin. And the hawksbill turtle is actually critically endangered. And then there's other portraits of organisms that aren't necessarily endangered, but iconic or meaningful to me in one way or another, such as the poison dart frog. So this is one way I sort of express that passion for biodiversity, is by trying to fantastically, so to speak, immortalize them through art, but also try and support them through wildlife conservation. But then on the science side, what I like to do is try to understand the processes that can result in biodiversity. And namely, I focus on the tempo of evolution with a even more of a focus on rapid evolutionary processes. So there's so many different kingdoms represented in this picture. I particularly like to focus on the fungal kingdom. And the fungal kingdom is a real Jekyll and Hyde type of kingdom. A little bit of just some base facts about fungi. They're more closely related to animals than plants. Some of them are genetically manipulatable. And so given some of their natural talents and some talents that we've bestowed to them via genetic manipulation, they produce great things for humans, such as beer, wine, bread, biofuels, rubbers, even components of Legos are made from fungi. But then they also have that flip side. And they're responsible for the death of over 1.5 million people annually. So part of the reason that they are such a two-faced kingdom, so to speak, is that there's so many species. There's over 5 million species. And so what are some of the processes that have made such diversity in the fungal kingdom in both function and form? And so I've studied two events that have led to rapid evolution or contributed to rapid evolution in fungi, some of them recent events, some of them more ancient. And I've given talks at Vanderbilt on other platforms about a hypermutating lineage of budding yeast. So today we'll set that story aside and focus on a recent hybridization event, a recent rapid evolutionary event that has to do with the hybridization of fungi. So I think hybrids are probably familiar to most, but just to go over a very iconic hybrid, the mule is one of the most iconic hybrids because it's been able to bridge physically distant cultures and help humans acquire physically distant resources. And that's in part due to a phenomenon called hybrid vigor, where the mule has the best parts of both parents, at least in some sense. From the donkey, it has strength, patience, and endurance, and then from the horse, athletic ability and speed. And so part of the reason that this hybridization event is even possible is that despite some phenotypic differences between the donkey and the horse, genetically speaking, they're less than 1% diverged from one another. But of course, today we'll be talking about fungi. We'll be talking about two fungi that came together and formed an entirely new and viable species. So it's very distinct from the mule, which is an evolutionary dead end. And with this, I introduced the very sciencey part of the talk, when two become one. And this is all about hybrid origin of clinical fungal isolates, which can be read further about in current biology. So the sciencey part of the talk has three components. I'll first introduce the players, these Aspergillus fungi, and then we'll talk about the genomics of the hybrids as well as phenotyping of the hybrids. So Aspergillus fungi, they're pretty deadly fungal pathogens. They pose serious clinical, economic, and social threats to humans. And that's in part because of the 200,000 plus Aspergillus infections that occur every year. But to say all Aspergillus species are pathogens would be very misleading. In fact, only some species are pathogens, and those are the ones indicated by the maroon circle, which are known to cause disease in animals, including some humans. And so instead of being condensed to just one part of the evolutionary history or one part of the phylogeny of Aspergillus species, they're kind of diffused throughout, suggesting that pathogens have evolved multiple times amongst Aspergillus species. And so I'm a graduate student in the Antonis Rokas lab, and one of the questions that as a group we're trying to address is what are the evolutionary forces that enable some organisms to be pathogenic while others are not. So we've focused on a couple of different species, one of which is called Aspergillus nigilans. And it primarily infects those with a disease shorthand called CGD, or chronic granulomatous disease. However, this species has also been associated with other lung-related diseases. So to try and understand why this species is causing disease, we decided to examine those isolated from patients. Specifically, we leveraged an international network of collaborators that were working in the clinic and were able to extract these isolates from patients' lungs. They identified them in the clinic, again, by clinicians, as Aspergillus nigilans. And from there, we also conducted genome sequencing, hoping to find the needle in the haystack of what was causing their virulence, or at least have some sort of clue. But upon genome sequencing, we realized that these were not the typical Aspergillus isolates that we've been studying for many, many years. A typical Aspergillus genome has around 10,000 genes and a genome size of 30 megabases, and that's what's depicted on the X and the Y axis. But these genomes had twice of everything, and these are the ones shown in purple. They had twice the genome size, twice the number of genes, and on the Z-axis are how many gene duplicates they had, so how many times did their genes come in twos. And so this led us to believe that these weren't at all Aspergillus nigilans, that they were something else. And so we turned to molecular phylogenetics to determine what these species were. But as noted in this figure, they had two of everything. So when we conducted molecular phylogenetics, we found that one copy of a gene tended to fall in one clade, which is depicted in red, while the other copy of that same type of gene fell into a blue clade. And so just to show you where two copies of one gene came from one isolate, I've highlighted here clinical isolate ASFU 2033. And it has two copies of one gene that fall again into two distinct clades. So this suggests that they are, in fact, hybrids between two distinct species. So to cartoonify what we think happened, to put forth a model of how we think these hybrids formed, we think that two distinct species were at some point living very physically proximal to one another, and they underwent something called cellular fusion. So now in one multicellular filament, there's multiple types of nuclei. And then we suspect that thereafter there was nuclear fusion, which created an entirely new species depicted in purple, and it now has two sets of everything, one from each parent. And unlike the mule, as I noted before, these are completely viable. They can produce both asexual and sexual spores, suggesting that this hybrid event is not an evolutionary dead end. Okay, so now that we have the genomes of these hybrids, we wanted to next conduct various analyses on them. But it's very helpful to know which genes came from which parent. However, they're all coming in sort of a mixed bag, and so we needed to find a way to tease apart which gene came from which parent. So we conducted this analysis that allowed us to do just that, based off of calculating sequence divergence of each gene to a known parent. So what you do is you plot the distribution of sequence divergence along all genes, and what you'll find is there's going to be one set of genes that are very similar to that known parent, and so they have very low sequence divergence, while there's going to be a second set of genes that are very different from that parent, and so they have higher sequence divergence. And you can look at other properties thereafter, such as how many of these genes are represented in like the total, and that can be viewed through a pie chart. So what we found in our hybrids is that both parents are roughly equally represented. We were able to pull out the blue genes in all of the hybrids. We were able to pull out all the red genes in all of the hybrids. And just looking at the pie charts, we could see that they're pretty much half-half. And just to highlight again, that same strain that I highlighted earlier, that we have strain ASFU 2033 with that perfect bimodal distribution, and roughly half of the genes are blue and half of the genes are red, or half of the genes are from one parent and the other half from the other. So now that we've sort of mapped out which genes are from which parent, this now allowed us to do a lot of other analyses, such as looking at the stability of gene pairs, looking at recombination. Through other means, we examined copy number variants and aneuploidies. And all of this indicated that these hybrids were very genetically stable, which is unlike the typical hybrids that you see, where having two genomes in one is a bit of an overwhelming biological scenario. And so typically hybridization is followed by genome decay because it's a very unstable situation. So again, we have this new hybrid organism that is the result of Aspergillus spinulosporus depicted in blue and Quadrilineatus-like species depicted in red. And what we think is even more remarkable about this genetic stability is that these two parents are very different from one another. In fact, they're about as different as a human is to a lemur, or roughly 8% diverged from one another. And this is again remarkable considering the mule hybrid that most of us are very familiar with, the parents are less than 1% diverged. Moving on to the last section, which is phenotyping of the hybrids. I think the biggest take-home message here is that the hybrids exhibited pretty wide phenotypic variation. So we examined a lot of different types of phenotypes that were all associated with virulence, such as virulence itself, which is the ability to kill a model organism. We looked at the interactions that the species had and that the strains had with human immune cells. We looked at antifungal drug susceptibility, et cetera. And we could summarize all of that information using principal component analysis. And so again, the hybrids are in purple. They're pretty diffuse along that first dimension. And you can also see that they're more like one parent in blue than they are to their other parent in red. Lastly, I would like to highlight that they're very different from Aspergillus nigilans, which is what they were originally identified as in the clinic. And Aspergillus nigilans is that dark gray dot in the bottom left panel or quadrant of the principal component analysis plot. Just to show you what one phenotype looked like that really exhibited quite a lot of phenotypic variation. When measuring their virulence in a moth model, we found that Aspergillus latus strains were both the most virulent as well as the least virulent out of all of the ones that we had looked at. So there's substantial heterogeneity. Amongst other traits, we also noted that some were more like one parent compared to the other. So for drug susceptibility, the Aspergillus latus strains seem to be more like their blue parent, Aspergillus spinulosporus. But when it came to resistance to hyphal killing by human immune cells, they were more like their red parent, Aspergillus quadrilineatus. And so we suspect that this might be something very similar to what was observed in the mule, where it has great traits, so to speak, from both of its parents. Or more specifically, we hypothesize that these could be signatures of hybrid vigor. And this is something we hope to investigate further later. So again, this manuscript was published under the title Pathogenic Allodiploid Hybrids of Aspergillus fungi in Current Biology, and further details can be found in that manuscript or summarized in the graphical abstract. But I think it can be distilled even further than that into two main points. Medically, we show that it's really important to be able to accurately identify the isolates that you're trying to manage the disease of. And then strain heterogeneity is very, very important. Evolutionarily, we suggest that hybridization is a general mode of fungal pathogen evolution. So they weren't really observed in these types of fungi before. And this, to our knowledge, is the first case. So with that, I would like to thank the project team, for which there are many people. We were led by the advisors Antonis Rokas and Gustavo Goldman, who's in Sao Paulo, Brazil. And the groundwork was co-led by myself and a former graduate student at Vanderbilt University, Abigail Lind. I would like to also thank the entirety of the Rokas Lab, HHMI for their funding of my predoctoral work, as well as everyone in attendance for your attention. Thank you.
Thank you so much, Jacob, for a wonderful talk. Again, if you have questions, please type them in the Q&A of the Zoom meeting. I'll start with a question. You mentioned that these results show that, right, hybridization is more general, perhaps, than previously thought among fungal pathogens. So can you elaborate on that and tell us a little bit about what is known about hybrids and in the context of fungal pathogenesis?
Yeah, so there's a handful of hybrids that are known to cause disease of sorts. They're typically like plant pathogens for the filamentous fungi, or for yeasts, they're pathogens of humans. So Cryptococcus neoformans, which causes cryptococcosis. There's been known hybrids there, as well as in some Candida species. However, for filamentous fungal pathogens for humans, hybrids had not really been seen, especially these allodiploid hybrids, where they really have the entirety of both parents rather than like a admixture type of or introgression type of hybridization event. So these are what differentiate, these factors are what differentiate this type of hybrid from other hybrids.
Thank you so much. And you know, if I can follow up, what do you think is the sort of next step for the project? Where do you see this going? Yeah,
I think that so the take-home messages, there were two, right? One was medical, one was evolution-based. And I think that it will really depend on like how you want to choose your path. There's the medical route of trying to understand more about virulence and its pathogenicity, as well as its prevalence in the clinic and how frequently these isolates are getting mistyped. And then evolutionarily, there's, I think, a lot of questions that are open-ended, such as, are they really as stable as all of our analyses indicated? And what is the life cycle like? You know, how many rounds of post-meiotic mitosis are there? Questions like that are going to be fruitful for understanding how this species is okay with being a hybrid, a human lemur equivalent.
And then, do you have any thoughts on, right, the comparison between human and lemur being 7% divergent, right? And you would never imagine hybrids there, even if there was sort of physical compatibility was not an issue. But fungi seem to be having no problems in terms of the plasticity of their genomes, for lack of a better word, in creating these hybrids. And so, do you have any thoughts on, you know, what might be making fungi so flexible?
Yeah, I think, like, I think that we don't see hybrids everywhere in the fungal kingdom because their lifestyle might not be amendable to it. However, the clade that the close relatives, the parents, are from, they're in a part of the phylogeny that, or they have life history traits that make them more amendable to a hybridization event, where there's rampant sexual reproduction versus asexual reproduction. And also the cellular fusion and having multiple nuclei just in one cell, it's kind of a common thing amongst some fungi, these ones included. So I think that those things make them more amendable to a hybridization event because they're kind of used to having multiple genomes. They're kind of used to the cellular fusion process, and all of these things kind of set the stage for making a hybrid far more likely.
Great, thank you so much. I would like to thank both Jacob again, as well as Stephen, for two wonderful presentations, and then invite all of you on our social afterwards. We'll start in about 15 minutes, and I believe all of you have received an email with instructions on how to connect to that Zoom call. So thank you so much, and have a great day.